The ligand binds to the G-coupled receptor. The receptor then becomes activated and its shape is altered. The receptor's cytoplasmic side then activates the G protein by turning the GDP into GTP. The G protein then diffuses across the membrane to bind to the enzyme adenylyl cyclase. Adenylyl cyclase then converts ATP into a second messenger called cyclic AMP. CAMP then diffuses across the cell to activate a protein kinase in this certain case. The ligand, which is shown by the red balls, binds to the tyrosine kinase receptors. This causes ATP to be activated and the tyrosine kinase receptors to close together. The ATP is then phosphorylated into 3 phosphates on each side, and ADP is shown as the orange balls. The phospholipase C then is activated. The phospholipase C then activates IP3, which then goes on to activate the calcium channels to open up and let the calcium ions out of the endoplasmic reticulum. The calcium ions then go on to activate an enzyme.
In a normally functioning neuron, there are receptor sites for epinephrine, which is released when the body is under high stress or pain. This causes the release of dopamine. However, when heroin is released into the blood, is released into the brain, it replaces the epinephrine and releases dopamine to the cells. This causes euphoria to be, or the feeling of being high to be experienced by the user.